Hello everyone. My name is Pulak. I'm the scientific director at Union Biometrica. And today I'm going to tell you about our flow cytometers and how they can be used in the plant biology field. So I've structured my presentation in two parts. First, I'll describe how our large particle flow cytometers work, pointing out some of the key features. And then I'm going to present some examples of applications using plant samples of various types, including pollen, seeds, protoplasts, and calli. So Union Biometrica makes large particle flow cytometers, and we have three different platforms: the COPAS FP, the BioSorter, and the COPAS Vision. Most of the data that I'm going to tell you about, actually all of the data I'm going to tell you about, was collected on the COPAS FP and the BioSorter. But the only reason we don't have COPAS Vision data yet is because it's our new instrument and we just haven't quite gotten a chance to collect data on the COPAS Vision, although because of the similarities, we expect that the data will be very similar and for some applications probably even better than what is being currently that we can collect on the COPAS and the BioSorter. What are the benefits of flow cytometry? I'd like to first of all point out that flow cytometers are high-throughput microfluidics devices that objects travel a known path so that you know where to look for them and to take measurements. What are the benefits? What's the big deal about flow cytometry? First, and there are a number. One is that you can take multiple measurements of each object in the sample and you can interrogate every object. Because it's a machine that's making those measurements, you're reducing or eliminating any bias that might be introduced by the researcher. So you're getting an unbiased analysis. Because you're able to take collect large data sets, take many measurements of many objects in your sample, you can start detecting small differences that are statistically significant, something you might not otherwise be able to do. And finally, and maybe the most important feature of flow cytometry is that it provides an increased throughput for handling this size of object. So to do an experiment with these tiny little objects like pollen or protoplasts or any other small object that is normally analyzed by flow cytometry, it would be very tedious to set up those kinds of experiments. And if you have an analyzer that can also sort, you can set up experiments that you otherwise would never even consider trying. On this slide, I would like to describe the analysis and sorting principles of our large particle flow cytometer and sort of describe how the instrument works. And I'm going to start by telling you what we mean by large particle flow cytometer. So this is a flow cytometer, and traditional conventional flow cytometers were initially developed as blood analyzers. So they can work with sample types that range in size from a little less than a micron up to about 40 or 50 microns. For our flow cytometers, we can work with a much broader range and much bigger sample type. The bottom end of our range is around 5 to 10 microns, but can go up to 1,500 micron wide diameter objects, much bigger than what traditional flow instruments can do. This is a little bit of a soft number, this 5 to 10 microns at the bottom end, because it sort of depends on on the sample type. We know that sort of the beads that are used by traditional flow cytometers to standardize and set up their instruments that are as small as 2 microns, we are able to detect these 2 micron polystyrene beads. However, a polystyrene bead is not a cell, And so we kind of err on the side of caution and know that we're in this size range at the bottom end and can go easily up to 50 and can go up to 1500 micron diameter. The kinds of measurements we make are size, optical density, and multiple fluorescence measurements. Our instruments have 4 different flow cell sizes. And when I am referring to the size, I'm talking about the microfluidics passageway through the flow cell that the sample travels. And the 4 sizes of that passageway are, and it's a square passageway, and it is 250 microns on the side, or 500, 1,000, or 2,000 microns wide. I told you that we have 3 different platforms, the COPAS FP, the COPAS Vision, and the BioSorter. And I sort of said on this slide that the BioSorter has certain flexibility that the COPAS doesn't have. And that is because with the COPAS, you have to choose one of these flow cell sizes, and that's a fixed flow cell for that instrument. The BioSorter has this feature of being able to more easily swap out these flow cells because it's set up to be able to do that. And we have these fluidics and optics core assemblies, something we've given the name FOCA, that either has the 250, the 500, the 1000, and the 2000 flow cell, and can easily be swapped in and out of the instrument. And this allows the operator to select the most appropriate size for the sample type that they're analyzing. So very often this is in a core flow facility or with a group that is planning on sharing their instrument with many other labs that are going to work with all kinds of different sizes of sample types. Like other flow cytometers, we use multiple lasers. The standard laser, standard configuration is a 488 nanometer blue laser. That's the workhorse for flow cytometers, and it is for us as well. One of the features that I'd like to tell you about is the profiler, and I'm going to explain that in the next couple of slides. But this is a feature that is available for our flow cytometer because the objects that we're analyzing are larger, and so we can start collecting positional information, and you'll see that when I describe what the profiler is. The final point on this slide is that this is not only an analyzer, but it has a sorting mechanism. It is capable of sorting or dispensing from that sample, parsing out some subpopulations from that sample into wells and multi-well plates, and it does that in gentle ways that you can sort out live biological material and maintain viability. On the right, I have a schematic that sort of describes, how this works, how our flow cytometer works. I'm going to zoom in a little bit so that we can see this a little bit better. And we use a sheath reagent and push that through the flow cell to set up stable laminar flow, and that's pushed through by pressure. And when you have stable laminar flow, when sample is added and objects in your sample are added to that stream, they're carried at a constant velocity through the flow cell, and hydrodynamic focusing tends to bring them to the center of the flow cell. So if they are moving at a constant velocity, then as they pass through this laser, so they pass through the flow cell and they'll pass through one or more lasers. And in line with one of those lasers is a detector. And as the object passes through that laser, it casts a shadow on the detector. And the length of time that it casts the shadow is proportional to the width or diameter of the object that is in your sample. In this case, there's small little green and red balls, but they could be pollen, they could be seeds, they could be other sample types. So one of the measurements that we collect is the size measurement that we call time of flight, to reflect that it is a measurement of the length of time that it took to fly through the laser. And since it's stable laminar flow, it's proportional to, that time is proportional to size. So it's a relative size. We also collect an optical density measurement off this detector, and that is a measurement of how much of the light passes through, an integrated value of how much of the light passes through the object as it's going through the laser. And so that's two of the measurements. Orthogonally, we collect fluorescence data to multiple fluorescence detectors. And in this case, they're photomultiplier tubes. And in the standard configuration, it's something in the green part of the spectrum, the yellow part of the spectrum, and the red part of the spectrum. But this is all controlled with dichroic mirrors and bandpass filters, so you can focus on whatever part of the spectrum is most interesting to the sample type that you're analyzing. So once the object goes through the lasers, and essentially as soon as it leaves the lasers, because all of this is happening at the speed of light, we already know that this is a green or a red dot, a red ball. We already know that answer. So it happens very quickly. And if it is an object that meets our sort criteria, we can send a signal to this air diverter that there's something coming that we want to collect. And the air diverter turns off and then on again to generate a slice of liquid with the object that we want to continue downward and be collected in the receptacle of our choice, like a well of a multi-well plate, or in bulk, or in the case of Arabidopsis seeds, maybe to an agar pad so that they can germinate on that agar pad in a specific location, in a specific pattern in a specific location. So that's kind of the kind of thing that can be done. So this really describes the entire process, how the analysis occurs and how the dispensing occurs. And that's true for all of the three different platforms and for all of the flow cells. I told you I was going to tell you about Profiler, and I told you that it allows you to collect positional information. And I'm going to explain it to you using hydrogel beads, and in this case, alginate beads. And they're often used to encapsulate either cells or cell clusters or something else, and provide a structure for those cells. And that's actually, there are a lot of researchers that use that approach with hydrogel beads. But I'm just going to do this for the purpose of describing what a profiler is. So if one of these beads, and we see a whole bunch of them sitting here, they're all empty, nothing is in them. If one of them goes through, the leading edge causes some kind of a leading edge effect as the laser hits that edge. And then there's kind of a low level of extinction signal, optical density, light scatter that's happening as the bead passes through the laser. And then as it leaves, again, there's this edge effect. Now, if there's something inside the bead, and I'm going to do this again. And so there's the outline of the bead, and within it is a cluster of cells. And if one of these now goes through, again, you see that edge effect as the bead first goes through, and you're taking multiple measures and plotting those multiple measures sequentially. Now, as the cell cluster goes through the laser, there's an increase in this case, in the extinction signal. And then as the bead continues further, the signal drops back to baseline, back to the baseline of the bead, and then there's the edge effect as the bead exits. So now we can get a measurement that is the peak height, the peak width of the cell cluster, and if there were multiple cell clusters in this bead, we might see peak numbers that we could count and relative position. This particular bead went through such that that cluster was more or less in the middle. This image has the bead at sort of at one end, and this is just a representative of the kind of beads that hydrogel particles with cells in them that were analyzed. Okay, so that's the profiler. And this measurement that you see here is for the optical density, but you can also get the same measurement for fluorescence in all of the fluorescence that you're measuring. So there could be like 3 colors of fluorescence, and that greatly increases the number of measured parameters for each object and increases the information density for each object. So the quick overview and the summary of the technology is that we have these 3 platforms that you can take multiple measurements, interrogate each object, unbiased analysis, large numbers analyzed, increased throughput, and automation to work with these small, tiny little samples. So that's the overview of the technology, and now I'd like to turn to some examples of data that we have collected. And the first example is, and I'm going to spend a little time on this slide, is with Chlamydomonas. And this is Chlamydomonas on a 250 micron flow cell, or FOCA, in this case it was a BioSorter, with the 488 laser, and one of the fluorescence detectors has a bandpass filter in front of it to collect the fluorescence that we know comes from chlorophyll, around 700 nanometers. So from one of our detectors, we're getting measurements of chlorophyll. In this experiment, we also have thrown in cytox green, which is a live dead stain that binds DNA. It's excluded from live cells, but can enter cells that are sickly and are dying. And then, so the DNA is taken up, binds to the, or the stain is taken up, binds to the DNA, and now those cells express a green fluorescence. The experiment was, we analyzed them, we sorted out live ones, we sorted out dead ones on the basis of the green and the red fluorescence, and verified it by microscopy. Now, how does that experiment look? How do you do that experiment? You do that experiment by running some of the sample through first, and you are collecting measurements from the Chlamydomonas, and they are showing up as red dots on these dot plots. And so I've shown on this slide 2 dot plots. One where I'm looking at the plotting of values of time of flight versus the red peak height, and also looking at the very same data on a time of flight versus green peak height. So we run these Chlamydomonas through, these red dots start appearing and they start accumulating, and in a couple minutes, we quickly have 2,000 red dots on one of these dot plots, and also the same number on this dot plot because it's the same data. We then notice that there's a cluster here and a cluster here. And on this dot plot, there's a cluster here and a cluster here. So the operator draws these regions around those clusters. So we call this one cluster R1, this one R5, because R2 and R3 are on some windows that we're not showing you. On this plot, we draw a region around, a trapezoidal region around here, or rectangular region around here, trapezoidal region around here. And now we want to figure out what's going on here. So I'll tell you that you can select a dot, a Chlamydomonas that went through and was measured in this window, and find the corresponding red dot in this window. And when you do that, you see that those Chlamydomonas that have measurements that put them within this cluster end up on this plot being in this cluster. So the high red, so this is a time of flight versus red peak height. The high red has low green fluorescence. Although it says GFP, it's really green fluorescence from cytox green, not green fluorescent protein. So those Chlamydomonas that have high red have low green. Those that have lower levels of red have high levels of green fluorescence. When you take a look at a representative profile from one of these in this region, and it's the same profile as one of these in this region, you see that you have essentially no green fluorescence, but you have this red fluorescence through that chlamydomonas as it went through. And most likely from the chlorophyll at around 700. If you take and look at a chlamydomonas that fell within this region here, which is also this region here on this plot, and you take a look at the profile, you see that there is a lower level of red and some green fluorescence. And we know that cytox is taken up by cells that are very sick and dying. And so these are likely to be dead, and we can sort them out, look at them under a microscope to see if they are showing signs of death or life and verify that. And that's what we did. And as expected, these were alive, these were dead, and you can sort on this region. You can use this as a sorting region, or you could use this as a sorting region. And similarly, this or this is a sorting region. So that's kind of an explanation of what these look like and what you're looking at. And you're going to see in upcoming slides more of these dot plots and profiles, and that will help you interpret them. The next example I'm going to show you and talk about is Sataria protoplasts. So these are protoplasts from foxtail. And again, it was on a 250 micron flow cell with a 48 laser. And what we see when we run those protoplasts through is a fairly uniform time of flight. Everything is falling within this size range. Time of flight is on this axis. And then this is the log scale for green fluorescent protein. And these protoplasts have been transformed with a plasma that expresses GFP off of some promoter that wasn't shared with us. So it's expressing GFP off of some promoter. And we see that there's kind of just a distribution from high down to low levels of GFP. And so we arbitrarily selected and drew a region around the highest fluorescence, a region around the lowest fluorescence, and some region in between. And then we took a look at the profiles, and we sorted these out singly so that the researchers could take these to a fluorescence microscope and take an image, and we have the profile that corresponds to that particular protoplast so that we could correlate the image that they got with the profile. And this is what we're showing you here, that this particular protoplast that was high green also has in the profile a very high green signal. This with an intermediate level of green has a green signal that happens to track perfectly. It happens to track perfectly with the extinction. So the blue signal is the optical density or extinction signal, and here they're just overlapping perfectly. Further down, the ones from the lowest green fluorescence have no green signal in the profile, nor do they appear to have any green signal in their confocal image. I'm going to point out to you that these protoplasts are still showing, we're still measuring some low level of green. It's not down here, not at the baseline, it's not zero. And that's because there's always some autofluorescence present in the green and in the red, and different levels of autofluorescence in different kinds of biological tissues. So that should not be a surprise, that even though we don't see any green fluorescence, we're still measuring some low level of green fluorescence, of some low level of green autofluorescence. Okay, the next example I'm going to show you is for fungal spores, and this is collected trichum of fungal spores. And these are fungi that can be plant symbiotes or phytopathogens of other plants. So they are of biological interest to people studying plants. A sample of spores were given to us, and we ran them on a 250 micron flow cell. Because they are small, so we ran them on our smallest flow cell, used the 488 laser, and then took a look at what the data looks like simply on the time of flight versus extinction. And you see this kind of distribution of signals. And if we zoom in, we see that this area that's blue, what happens is that each red dot is a single event. But when you collect here, we've collected 9,000 signals. Many of them have multiple copies with the exact same values of time of flight and extinction, which is not a surprise, right? The spores are all about the same size. So a whole bunch of them are now getting the exact same measurements and are landing on top of each other. So we know that this is the sort of predominant, most common time of flight and extinction size. So we drew our gate region around that and then sorted these out one at a time, and then had the researcher take a look at them under a microscope, and sure enough, there is what they look like. And you see they're not round, they're kind of elongate, and that is a sample type that we are also able to work with. They don't have to be round objects. They can be rod-shaped, they can be elongate, they can be all kinds of shapes. They don't have to be round. Next example is Arabidopsis pollen. And I think this is a good example to point out the strength of using this as for phenotyping your sample. So here is Arabidopsis for that, 4 different lines of Arabidopsis: a wild type line, one that is expressing GFP off of the LAT52 promoter, and the LAT52 gene is highly expressed in pollen. So this particular line has GFP expressed off of that promoter. Another line that has the LAT52 expressing GFP, but it also has the UDP1B 3' UTR element inserted into the gene, so that now the GFP expression is also under regulation of that element. And that element is a negative regulator of translation. So that's going to give you that information now, and you'll see why that's important later. And the third line is a line that is GFP expressed off of the MGH3 promoter, and this is a promoter that's male tissue specific. Okay, if we take a look at the data, what we have in this first panel is we see that the pollen, the vast majority of the pollen is over here, and then there's other signals present which happen to be debris and other things that are present in the sample, but they have this low level of extinction. And if we sort some of them out, we see that they're not pollen, but here's where the pollen is. Now, once we've established a gate region for pollen for wild type, that same gate region is likely to be, it's likely that we can use that same gate region for these other lines, as long as these lines haven't affected the size of the pollen. And in this case, that was true. The pollen all remained about the same size, so the same R1 could be used for sorting out the pollen for all of these experiments. Okay. The wild type levels of GFP peak height fell within this region here, and that is your baseline autofluorescence GFP. Now, in the line that is LAT52 GFP, you see this big shift where the vast majority have now moved up from this region of R5 up into the region of R4. And so this, and when you take a look at profile, a representative profile from this region, you see that it has the green fluorescent peak. And as you would expect, as it should, because it has higher green levels. If you sort something from this region, you don't see that green fluorescent peak. And so again, it is suggestive that the level of green expression is much lower down here. the 1545 line, which has the 3' element in the 3' of the gene, you see now that an increase in the amount of protoplasts that fall within this gate region, in the region that is similar to the wild type region with very little GFP expression. There are still some that are expressing GFP, maybe at a lower level, and so the element doesn't completely shut off the expression, but it does dramatically reduce the expression. And then the third example, the third line here, we see very little, we see almost everything drop back down to the wild type levels of green expression, almost as if there was no GFP present. Some of the protoplasts, some of the pollen do have GFP expression, and that's because there's going to be some leakiness, and maybe those are going to be interesting for some reason. So what else could you do with this? Now that you're able to sort these out, maybe you could sort out these that are still having some level of GFP expression and do an experiment like RNA-seq and see what RNAs are being expressed in the pollen that still retain some of this GFP expression. Maybe that would allow you to understand what's happening in these pollen that allows allows a greater level of GFP expression that normally only expresses in male tissue gametophytes, which really are at present a very low level in Arabidopsis pollen. Therefore, you wouldn't expect much GFP expression. Okay, that I did not want to do. Sorry about that. The pleasures of learning how to do all this on webinars. Okay, my next slide is an example of moss protoplasts, and this is Fiscomatrilla patens, and this was run on a COPAS instrument with a 500 micron flow cell with 48 laser, and we ran both wild type and GFP and a line that is expressing nuclear GFP. And what you see here is that the wild type, here's the wild type protoplasts run, and you see very low levels of GFP. I'm gonna again zoom in. Very low levels of GFP expression on your plot of red fluorescence versus green fluorescence. So you are getting signal from the chlorophyll, but you're not getting, well, these are protoplasts. So you're seeing about the same level of red fluorescence in both populations, but in the population that has nuclear GFP expression, you have much greater numbers of protoplasts that are present in this part of the plot. And when you sort out the protoplasts from this population and take a look at them under a microscope, you do see that the nuclei do have green fluorescence within the nuclei. The next example is another fungal example, and it's not, fungi are not plants, but it gives me an opportunity to give you an example with hydrogel particles. And these are fungal spores that are encapsulated in alginate beads using an encapsulation device from Selena. And these spores form mycelia. And when we take a look at these hydrogel particles that have the mycelia growing within them over time, we see that the extinction, the light scatter, increases from time 0 or in comparison to the empty beads. Look at the 10-hour beads, more so in the 12-hour and even more so in the 14-hour incubation, where now the beads are retaining their size. They're not changing much in the time of flight. They are changing a little. I think that some of them are starting to break open as the mycelia overtake the alginate. But what you do see is an increase in the scatter. So this is an example of the kind of what you can do with something that's encapsulated, and then sort out or parse out those at different stages and do further research with them. On this slide, I'm showing you data from pine pollen that was collected from lake sediments for isotope analysis. And the sediment sample is cleaned up a little bit to give us a uniform size, and then run through a COPAS instrument with a 500 micron wide flow cell and looking at autofluorescence in the area of around 610 to sort out these pollen away from other material in that sample. And in this experiment, we were able to sort out 30,000 to 60,000, depending on the sample that we were working with at the time, 30,000 to 60,000 pollen per hour to use in carbon-14 isotope analysis for carbon dating. Return to another example with Arabidopsis, this time seeds, and this was simply to show researchers at a demo that we can sort out rare events. So they spiked fluorescent seeds within non-fluorescent samples, and we sort out the green fluorescent ones away from the non-green fluorescent when you had spiking down at about a 1% level. We also did it at 10%, and we also did it at around 0.1%. So we were able to look at spiking at different concentrations without any difficulty of sorting these out. So this would allow you to think about doing experiments where you're looking for a rare event and sorting that rare event out. I will point out that it's a lot easier to find a bright fluorescing particle in a background of non-fluorescing than vice versa. But if you were trying to set up an experiment where you wanted to look at a decrease in the level of expression, that is a very difficult experiment to do. We could draw gate regions where at the bottom edge of a distribution where most everything is expressing some fluorescent protein, and sort out those that are a little bit below or at the bottom edge of that distribution, and enrich for those that are starting to decrease in their fluorescence levels, or maybe have now extinguished their fluorescence expression. And that is the kind of an experiment that you would be able to do, sort out those rare events because it could be the product of a mutagenesis, or it could be a rare event that occurs in the population. And that would be something that would be very easy to do. Got a couple other examples. One is calli from Cassava. And these are cells, protoplasts that are growing in culture, and they form these cell clusters. We asked the researcher to pass these through a filter to generate a size that was smaller than the 2 micron wide flow cell. And then we ran these. They were expressing GFP at a low level. As you can see here, some had higher levels of expression, some had very low levels of expression. We sorted out those higher levels of expression, parsed them out, collected those. Those also happen to have a lot of expression in the red part of the signal, around 600, and that's the autofluorescence. But the researcher was more interested in the GFP signal that was at this low level in these calli. We run other calli that were smaller that we could run on a 500 micron foci on a biosorter, where all of them sort of could pass through the foci. And we ran some, then arbitrarily selected regions of that were high extinction and big, and some that were smaller still, and the smallest group. Sorted these out one per well, so that we had one per well and the profile that corresponded to the object that got sorted. And then the researcher collected images for us. I wanted to show these images because these are bright field images. And these are the kind of bright field images that the COPAS Vision collects. These are not images from COPAS Vision, but we are looking forward to getting a chance to do experiments with calli to be able to do something like this. And you can see, again, that you can collect calli of all kinds of different sizes and shapes. And that would allow you to parse out this, this population into calli of different sizes. And it's quite likely that cell clusters of different sizes are undergoing different kind of biology. So these can be collected, RNA-seq experiments done to determine what genes are being expressed here and compare them to those that are bigger. You could set up this experiment where promoters are getting turned on as the cells start to differentiate and start moving along the path to redifferentiate into a plant, and start taking a look at what genes are being turned on and see if there's a correlation between the size and what gene is being turned on, or have multiple promoters expressed at different, multiple promoters that are reflective of different physiological states, and take a look at how those are being turned on with respect to size and other growth conditions. So my last example is Wolfia, and I think this is pretty cool because I didn't know, but Wolfia has the distinction of being the smallest known flowering plant. And we got a chance to run this on a biosorter on the 2000 FOCA, the one with a 2000 micron wide flow cell. And look at it with 48561 lasers, as well as something that could detect the chlorophyll at around 700 microns, fluorescence from the chlorophyll. And we were able to keep the small little plants suspended in solution. And then we took a look at the fluorescence or at the size of the plant and the different levels of red peak height reflective of the fluorescence from chlorophyll. And we see that there's a whole bunch here up at the top. There's a distinct cluster here at the bottom, and then something in between. And this experiment also, or the instrument was also set up to pick up side scatter from the 488 laser. So I'm pointing that out because when we sort out from this region up here, we see that there's a lot of red signal, indicative that these are probably healthy, alive and healthy. And when you look at them under a microscope, they're green. So they look like the green plant. When you take a look from down here, there's very little red signal. And when you take and you sort out, they look white. And you also see sort of this higher level of side scatter, much higher than you see with those that have the highest level of red. So those up here have low levels of side scatter, down here, higher levels of side scatter. And then those that are intermediate seem to have an intermediate level of side scatter, but still very low or no red signal. So again, this now allows the researchers to parse these out and see what's going on here. What are these? What are these? What RNAs are being expressed? Do histoimmunochemistry to see what are the cell surface markers being expressed? How are these different from each other? And start characterizing them, or put them under different growth conditions. See if these can be converted into these. See if these get converted into these, and what biology is happening. All right, so that's a survey of the kinds of samples that we have run, and this is my summary slide. I told you about large particle flow cytometers, that our flow cytometers come with 4 different flow cell sizes, that there are, that the measurements we make are size, optical density, and fluorescence, multiple lasers for excitation, COPAS Vision, we can capture a bright field image. I did not show you data for that, but I want to let you know that this is something that I think will be of value for many types of experiments, that we can generate these profiles and use them for analysis and sorting. And sorting and dispensing is gentle, so that even protoplasts and calli remain viable, and that the collection can occur in many different formats, into multi-well plates, into tubes, to the surface of agarose pads that are being used to germinate Arabidopsis seeds, into whatever the researcher would like.